Hey everybody, this is our module 2B, and we'll continue our microscopic brain examination in this module. And before we talk about this module, let's talk about how to become smarter. So there are two ways that you can become smarter, at least from my perspective. First one is to Google deeper, and second is to use the tools that you learn at school. So Google deeper means to use your knowledge to assist in your Google. This is really a name that I invented. Now let's take a look at an example. This is one of the questions from our module 2A. I ask you those DAPI-stain nuclei might belong to which kind of cells in the corpus callosum, right? And then the knowledge that you should already have, you should know that DAPI-stained nuclei and corpus callosum belong to CNS and Schwann cell belongs to PNS. For the last piece of information, you may not remember that well, but you can use Google to confirm that. And now you can use Google more efficiently. You can Google the locations of an ependymal cell, and Google tell you, okay, they are in ventricles. And then you know, okay, ventricles is not corpus callosum, so this is not the right answer. And Google will also tell you that red blood cell doesn't have nuclei. Now you should know the answer that red blood cell is not a choice because DAPI stain the nuclei, right? So now you should appreciate why do I push you hard to remember all these crap things, like weird things, because the more things you know, the smarter your Google will be. Another way to get smarter is to use the tools that you learn at school properly. So here is the example. If I ask you the ACO, anterior commissure olfactory limb, and hippocampus, which one is more anterior? If you ask Google, Google will not tell you. If you're smart and you think, okay, let me Google an image of sagittal brain slice, and then plus the keywords anterior commissure, olfactory limb, and hippocampus, you will know the answer, but you can also use the tools that we learned at school, right? The brain atlas. If you search around the images, and you will figure out where the anterior commissure located, where the hippocampus located, and you know their relationship, right? Okay, let's go back to our lectures. So this is the objectives that we're going to learn this week. And particularly, we're going to talk about the histology. So histology is the study of the microscopic structure of tissues. And what are tissues? They are made up by three parts: the cells and the extracellular matrix, and then the body fluid. So an example of the matrix is the collagen. So when you're younger, you have a lot on your skin. So your skin looks shiny, looks watery. So when you get older, all these collagens disappear, and then you will look, oh, with a lot of wrinkles. And there's only four types of tissues in our body. Only four types of them. So the first one is epithelial, and then the connective tissues, and then the muscular tissue, and then the nervous tissue. And in the nervous tissue, you have two types of cells. Only two types of cells. One is the neuron, that is the something make you smart, make you the you are, and then the glia cells. They are the supporting cells. They do the feeding, they do the maintenance, they do the cleaning of the neurons. They sound like not important, but without them, your neuron is not going to work. Think about this. All the city workers, if they quit, this city will become a big chunk of trash. Is that right? Okay, let's talk about the procedures of doing histology. The standard protocol includes 5 steps: fixation, embedding, sectioning, staining, and imaging. And the embedding is depend on your need of your next step. Sometimes you don't have to do that. Okay, let's talk about one by one. First one is the fixation. So the purpose of fixation is to deactivate the enzyme activities in your tissue. If you are a dead body, if you don't do anything to the dead body, it's going to deteriorate pretty soon, right? So that's one of the purpose of doing fixation. Another purpose of doing fixation is to make the tissue a little bit harder, to harden the tissue a little bit. You remember when we're doing the sheep brain dissection, the sheep brain feels a little bit rigid, right? If we didn't do the fixation, first one, when the brain gets you, it will become pretty smelly, going to go bad, right? And also it will be very squishy. It's going to feel like a jelly, so you won't be able to dissect any distinct structure at all. There are 3 ways of doing fixation. So the first one is use the heat. It's just very similar when you're cooking meat in your kitchen, but it's not very commonly used now. And the second one is immersion. So immersion means you collect the tissue from a dead animal and then you dump this tissue into formalin. You just immerse that under some solution, and that solution is formalin. And then the chemical name for formalin is formaldehyde. Then the formalin will get into the tissue little by little as a kind of making a pickle. For this method, if the tissue is too big, the center part of the tissue might go bad before the formalin can reach there. And that is why when we're dissecting the sheep brain, some of the sheep brain don't have good structure in the center part. And to solve this problem, people invented another technique that's called perfusion. And usually people will use transcardial perfusion. So trans means through, cardio means heart. So for this technique, people will pump formalin into the heart. So once the formalin get into the heart, it will go into the blood vessel, and then the blood vessel will deliver formalin to every single corner of the tissue very fast to make sure that formalin can reach there before the tissue get bad. There are 4 steps of doing transcardial perfusion. First one is anesthetize the animal, which means you put the animal asleep, but you cannot kill them. And then you flush out the blood with PBS. For this step, you pump the PBS into the ventricle of the heart, and then you make a cut in the atrium, so the blood will get flushed out from the atrium. And then you perfuse formalin through the heart, or formaldehyde, which means you pump formalin into the ventricle of the heart. So this way the formalin will get into every single corner of the tissue, and then you collect the tissue that you need. Okay, here comes the question. Why do you have to anesthetize the animal? Can you kill it? The answer is no. Why? Because when you kill the animal, the blood will clot. If your blood is clogged, then you won't be able to flush the blood out. Blood vessel will be blocked. You won't be able to deliver formalin to every single corner of the tissue, right? Okay, here are the photos how we did transcardial perfusion on the mouse. So first one, we inject some drug to the mouse, so this little guy will fall asleep. And then we cut the chest open, and this is the heart. So we will insert a needle to the ventricle of the heart, and this needle was connected to the pump. So the pump will first pump PBS, so the blood will get flushed out because the atrium was cut, so the blood will flow out from the atrium. See, this blood are coming out, and then we'll pump formalin into the heart. And then when the formalin gets into the tissue, the mouse will become rigid. Their tail is going to be a stick. And then you are going to cut open the skull and pop up the brain. And this is the brain that you collected from the transcardial perfusion of mouse. Okay, and the next step is embedding. So the purpose of embedding is to make the tissue harder. So the harder the tissue, the easier to thin slice it. So if you have been cooking in the kitchen, then you will know a frozen beef is easier to cut thin than a room temperature beef, right? So that's why we have to use this embedding if we need to have very thin slice. So if you need to cut very thin slice, like less than 1 micron, 1 microliter thin, you are going to use resin to embed your tissue. And if you need 5 to 10 micron, you will need a paraffin, which is a kind of wax to embed your tissue. And this is how the paraffin embedding looks like. So the white part, that's a wax. And you can also use freezing. If you froze the tissue, you can make a wide range of sections. And if you need a thicker slice, like the slice that we use in our lab, which is 50 micron, you don't need to embed. The fixation, the formalin fixation, will harden it enough so we can make a 50 micron slice. And then you can section in your tissue, which means to thin slice the tissue that you collected. And you have 3 choices of equipment. The first one is the vibrotome, and then the microtome, and then the cryostat. Let's take a look at them. And this is the vibrotome. The vibrotome will have a chamber and have a blade here. And this is a microtome. So the vibrotome also has a blade, but Vibrotome we usually use for paraffin embedded tissue. You see here, that's the darker part, that's the tissue, and then the white part, that's the paraffin, that's the wax. And then when you're trying to cut it, you are going to have a ribbon of your slice because the adjacent slice will stick to each other. Then you're going to have a long slice coming out from this cutting. And then the cryostat, that one also have a chamber, but this chamber will have very low temperature, so you can cut frozen tissue in this cryostat machine. And also you will have a blade here. Okay, so this is the Vibrotome that we have, and I use this one to make the slice that we stain in the lab. And you can see there is a spring here, and this spring was glued to this platform, and behind this spring there is a piece of agar gel. And this agar gel will prevent the knife from hitting this metal protector. And then this is the blade. It will vibrate when it cut. And when you're cutting the tissue, you are going to fill up this chamber with PPS. So every time you get a thin slice, the slice will float in the water. So it will be easier for us to pick up the slice. And then the blade will go forward to cut the brain tissue. So after the cut, the blade will go back and drop 15 micron. And then the next time when the blade comes to this way, it will cut a slice that's exactly 15 micron thick. If you want to get 40 micron thick tissue, you can adjust the blade to a drop 40 micron. So you'll get a thinner slice. And then the next step is staining. So we're going to talk more about the staining in the next slide. And then the last step is imaging. So you already know how to do that from last module, right? Okay, let's talk about the different staining techniques. So the goal of staining is to provide contrast so we can see small structures a bit easier. And there are 3 types of stainings. First one is classic histological staining, and we usually call this histochemistry. Histo means tissue, and chemistry means we are using some chemical, which is the dye that we use to stain the tissue. So histochemistry staining. And then in this type of staining, we'll use a dye to bind to specific structure. For example, DAPI stain is a histochemistry staining. So DAPI will stain DNA, particularly the AT-rich region of the DNA, right? And the second type is immunohistochemistry, the IHC. For this type of stain, we'll use antibody-based staining. We're going to talk this next week. And then the third type is RNA DNA-based. So in this situation, we'll label RNA or DNA with a dye. And this RNA or DNA, we call them probe. And then this probe will label DNA or RNA in your tissue. Okay, now let's talk about the dyes that are being used in histochemistry. There are 2 types of dyes, pigment and fluorophore. Doesn't matter which type, as long as it is a dye, it has to bind certain molecule. If it doesn't bind, you are not going to stain anything with them, right? Okay, so all the dyes have their favorite molecule to bind. Now, here comes the old question. What's the difference between a pigment and a fluorophore? Okay, so let's talk about specific dyes. First one, the DNA dye, DNA staining. We already used the DAPI last module, so you all know this is the DAPI. DAPI will stain the nuclei, right? And there's another type of dye, which is Hotchkiss, that works very similarly to the DAPI. It will bind to DNA, and then even the color looks very close to that. And there's another type of dye can also stain DNA, which is hemotoxylin. So this one is toxylin, hemotoxylin. If you cannot remember this word, you can just say this is the H dye. Okay, now compare with these three stainings. Can you tell which one is pigment, which one is the F word, fluorophore? This one is a pigment. Why is that? Did you see the background is white, is bright, and then the background of these 2 slides are dark. So if these 2 are fluorophore, that means you have to use our fluorescent microscope to see them, to see the result, right? But for this one, you don't need a fluorescent microscope. A regular light microscope will do your work. Okay, next one, eosin. Eosin is a dye that will bind to cytoplasma. It will stain cytoplasma. And eosin is a pink pigment. Sometimes if the stain gets really intense, it will become red. And usually eosin will use together with hemotoxylin to make an HE stain. So if you use the H and E together, you will increase the contrast even more to make your image more clear. Okay, let's take a look at the image that we got here. This is the HE stain, and this is the brain tissue. And then in the brain tissue, you have blood vessels, right? And this is a blood vessel. See the cell nuclei here? That is a nuclei of the blood vessel cell. And inside here, you see that very pink color. That is red blood cell. And then you learn that red blood cell don't have nuclei, right? So that's why you don't have any purple staining here. You only have this pink, almost red staining here. That's eosin stain. And next dye is Golgi staining dye. So this dye is a black pigment, which means this pigment will absorb all color light. No light escape it. And then you see a black in there, right? Okay, and then what is fancy about this Golgi stain is this stain will only stain neurons. The glia cells is not going to get stained. And what is even more interesting is this dye will stain a whole neuron like this, but it only randomly stains certain neurons. And which neuron get picked up, nobody knows. Now let's talk about Niso stain. The dye we use in Niso stain will bind both to DNA and mRNA. Where do you find DNA? In the nuclei, right? Where do you find mRNA? In the soma, in the cytoplasm of the neuron. Do you have any DNA and RNA in the axon? The answer is you are going to have a little bit mRNA in the bottom of the axon. So if you do a Niso stain of a neuron, the majority part that you will stain is the soma. You're going to have a little bit of axon stained, but not a whole axon. And how about the astrocyte? Are you going to be able to stain astrocyte using Niso dye? The answer is yes, because there is both DNA and mRNA in astrocyte. How about red blood cells? Can you stain red blood cells using a Niso dye? The answer is no, because red blood cells don't have nuclei, which means it doesn't have DNA. If they don't have the DNA, there's no way they can get mRNA, because the mRNA was made based on DNA. Let's talk about different type of Niso stain. Crystal violet is a type of Niso dye, and this dye is a pigment. And this is an image of crystal violet stain. Neurotrace is a fluorophore Niso dye. And this dye is a very quick procedure. We're going to use this dye in our lab. Crystal violet will take several hours to finish the whole procedure. Let's take a look at the image of a crystal violet stain. So this is the soma of a neuron, and then you can see this is actually the nuclei. And there's a structure called the nucleolus. This is a structure that when cells go to a certain stage, their DNA will condense into a small area. That's why you see a darker region here. That is the crystal violet stained DNA, and you see a lot of DNA in here. In the soma of the neuron, you see some dot, more purplish color there. That is Niso body. So Niso body is a rough ER cluster in the neural cytoplasm. So on a rough ER, you are going to have a lot of mRNA. That's why you have pretty dark stain in these regions. So they are Niso body. So you are going to find some Niso staining in the axon, but not the whole axon, just the starting part of the axon. So does dendrite. There is a little bit staining of the dendrite after the Niso stain. Okay, next stain is a myelin stain. There is a dye called Loxo-Fast Blue that will particularly bind to lipoprotein of the myelin sheets. Okay, so here is the double stain. The purple is the crystal violet stain, and this is under 4x objective length. So the dot, every single dot represent a soma of a neuron and maybe a little bit of the dendrite or the axon, but it's really hard to tell because this low magnification. And there's a blue color here. That is because there's a lot of myelin here. And as you already know, this is corpus callosum. Corpus callosum is white matter, and there is a lot of axon in there. So axon is myelinated, right? So that's why it is blue. There's a lot of Loxo-Fast Blue stain in this region. And so does this region. Actually, this is a fornix. If you remember, fornix is also white matter. All right, let's talk about the cortex. So cortex only find in mammals. Actually, some kind of bird, like songbird, they have very thin layer of cortex, and this cortex is really important because that is the brain tissue that makes you smarter than a worm. Okay, and then the cortex are organized horizontally into 6 layers. So you can see for a chimp, they have 6 layers, and each layer you can further divide them into A and B or even C. And so does the different like monkeys, tree through, they all have 6 layers. And the thickness of each layer varies in different regions of the brain. For example, the third layer is pretty thick in the prefrontal association cortex. This is some cortex located in your frontal lobe. And the third layer will become thinner when we moved to occipital lobe. This is the occipital lobe, right? And why do we differentiate the cortex into layers? Because their functions are different and there is different component in each layers. For example, when we are doing the DAPI stain, we have this first layer here. If you remember, the very bright layer is a meninge, that is not cortex. And our first layer of cortex is darker because there's less density of nuclei. And if you take a look at other kind of stain, for example, a Golgi stain, there's no stain here at all, which means there's no neuron in the first layer of cortex, right? Because Golgi will stain neuron only. And then if you do a nasal stain, you start to see this positive staining here, and think about what kind of cell they are. If you do a Viger stain, that is a stain very similar to Loxo-Fast Blue that will stain the myelination. So you see there is some myelination go this way in the first layer, which means there is some axon, myelinated axon go through this way. And in the brain, what cell will myelinate the axon? It's oligodendrocyte, right? So some of these nuclei will belong to oligodendrocyte. Is that right? Okay. And then the other layers, you can see the component of different layers are really different. That means different layers will have different functions. All right, that is the lecture part of this week. And in the next week, we're going to talk about the immunohistochemistry. We're going to do fancy stainings. We'll have different colors. And then we'll talk about the backfilling technique. And let's get ready to the lab.